Okay, and then we'll just give it a couple minutes for everybody to join and then we'll get started. Okay, we'll give it maybe 1 or 2 more minutes for people to join. And then I'll kick off the meeting and welcome the new members that we have as well if they were able to attend today.
Okay.
Okay, I think we'll get started. It's 9 o'clock. So welcome everybody. As I said earlier, Dan is off today, so you get the pleasure of having me to host for the first time. I just wanted to start by welcoming, we have 3 new members. So feel free to introduce yourself and tell us a little bit about yourself if you're online. So the first one is Jean-Christophe Ruel from Inoptech. I don't know if Jean-Christophe is here. I don't see him yet. So we'll just go on to the next one. And then we also have Ryan McCarty from Myconics. Is Ryan on today?
Yes,
Oh,
ma'am.
perfect.
Ryan
Yep.
McCartney here from Mycionics.
Yeah, so feel free to introduce yourself and let us know a little bit about you as well.
Cool. Thank you. I'm new with Mycionics in sort of the last 6 weeks or so. I'm a Waterloo Region tech guy, spent sort of 20 years building go-to-market functions for a couple of startups to scale up Smiovision and Axonify, and new to ag tech. Grew up in farming, and so wanted to get back to it and keen to be here and just appreciate the opportunity to get network
Perfect.
to learn about what everybody's experiencing.
Okay, great. Thank you, Ryan. And then our last new member is Howard Sinclair Jones. Howard, are you on online today? Just looking to see. Okay, I don't think he's on either. So we have 3 new members. So as always, if you know anybody who would be a good fit for the for our working group, feel free to send them our way, and then Dan or myself can connect them with signing up as a member. So I would like to welcome our presenter today, which is Chris. Uh, uh, by so sorry if I say your last name wrong. Bidinosti, from the University of Winnipeg, and he'll be talking to us about terabyte from data sets to data rovers and algorithms in between. So I'll hand it over to Chris to introduce himself and start his presentation.
Okay, thank you very much. Is that showing up?
Yes.
Okay, and let me just. Okay, thank you, everybody. Yeah, so there'll actually be three of us. I'll say a bit more. Here's the team. Terabyte's the name of our research group. It's myself. Chris Henry is at the University of Manitoba. I'm at University of Winnipeg. And Michael Beck, who's at University of Winnipeg as well, and along with our technician Tung, who's online. So I'm a physicist. Chris is a computer scientist. Mike's a mathematician who's now in computer science. But we've made our way into the world of agriculture, and we have with us a slew of students and technicians. So this right-hand figure I like. Everybody here on this call is here for that reason. There's just something very compelling about agriculture and applying what we can to it. And people have been thinking about these things for a very long time. Our way into this came from GPU computing, in particular through the NVIDIA architecture. So Chris and I got involved in that maybe about 15 years ago, and my interests were physics, Monte Carlo simulations for nuclear magnetic resonance, and Chris was very keen already at that stage on the machine learning. But it became clear that to us that there was a really interesting point in time, and maybe some of these things were coming close. And it was a good opportunity to try and move into digital ag, and we found sort of a niche that worked for us. So I'm going to talk about data generation, and then Michael and Chris are going to talk about two other aspects of our work. So we were very fortunate. Well, first thing when we got into this business, we discovered there wasn't much data out there. And so we decided we're going to make our own labeled data. And well, we've been fortunate enough to inherit a growth chamber, a big walk-in growth chamber. So this is back from photos from the COVID days, but we can grow and image things if people out there need that and label those images. Here was our first go at things. It's a gantry. Michael called it the Embedded Autonomous Generator of Labeled Images, or Eagle Eye. And what it is a three-axis gantry. And with, maybe I should put this laser on, with pan and tilt on the camera. So 5 degrees of freedom. We know the plants. We know where we put them in the volume. The camera wanders around and it takes images and it immediately puts a label on them. So we get immediately labeled image data. And we're up to over 1.5 million images, 18 classes, 56 subclasses, and those are all hosted and publicly available, as Michael will point out later. Some people might recognize this as a PhenoSpec multispectral scanner for plants imaging. It usually points downwards. We wanted to do wheat, so we rejigged it. We turned it sideways. We put it in its own sort of superstructure, another frame, so we can push it around and do whatever we want with it. And here's some examples of imaging wheat and wheat heads, and Chris will say more about those models later. Hyperspectral imaging, we got our hands on a few cameras. So, of course, you can extend the range of what you see with hyperspectral images. This has just led us. It helps us to start developing algorithms and thinking out how we can employ this extra information. Something I'm very keen on, low cost, very useful photogrammetry. So this is, a couple of Raspberry Pi cameras and a Raspberry Pi and a commercial turntable. But you can build up really beautiful 3D models. And again, Chris will say more about that. And the plant people really like this. It allows them to extract all the metrics that they kind of do by eye or by hand, but now you can do this objectively and with much more accuracy. We do outdoor imaging, so we collect for several years. We collected images in the field at Emily's Innovation Farm with and without herbicide. Here's a person driving the tractor collecting the data for us. So soybean, canola, and wheat. This is a photo of our colleague Maria Antonia's Fusarium head blight nursery at the AFC in Morden, Manitoba here. We do some drone stuff, but not much. But every year when it comes to rate her Fusarium head blight trials, she has a slew of students that go out with clipboards and by eye gives an assessment, a quick visual assessment. She can never get through all her trials, and if the weather's horrible, things can go awry. So we've been working slowly, trying to get ways to speed that up, and that's where the data rover comes in. So this is two models of it. This built mechanically by our friends Rob and company at Tech Industries here in Homewide, Manitoba. And so there's version one. We have a version two that's a lot more sturdy. It's a benchtop on wheels, essentially. And now I'll say a bit more in a moment. Presently, version two is over at PAMI. So I think everybody here knows the good folks at PAMI. And they're putting in the electronics for us and the brains into that. And presently, this is just a view of that fields at Morden. It's by remote control, which is good. Um, it's still, if we can get this up and running, we don't have to analyze on the fly, but even if we can get the data and the models and the video, we can collect that much faster than people can walk through the fields. And of course, we can make those assessments much better. We would love to have this to be autonomous eventually, but that I think is going to take us a while to get there. And maybe we need to pick a few brains of the folks that are on this call. So I'm going to kick it over to Michael to talk about our data curation and hosting. So everything, all the different imaging modalities, we post all this data publicly. But I'll say one last thing. When we got into this business, I was about 6, 7 years ago, I was very inspired by this paper. Image analysis in plant sciences publish then perish. And it was all about what we all know can happen. You have your work on a floppy drive back in the old days, or maybe then it became a CD, or maybe it became a hard drive. And then when the grad student leaves, it sits there collecting dust, it gets forgotten about, and all this lovely work just gets lost, or it just gets more and more complicated to keep track of it all. So proper curation was a big important aspect of our what we want to do right from the beginning. And now I'll pass it over to Michael, and I'm going to continue doing the slides, and Michael can share his insights on this.
Yeah, good morning, everyone. My name is Michael Beck, as introduced by Chris. So, yeah, as Chris already mentioned, data storage and data sharing is a pretty big deal. It might not be the most flashy part of our work, but it's pretty important nonetheless. So, what we see on this slide is basically the first setup of what we have. So, like all this data we are collecting, for example, through Eagle Eye is being stored at our university at a big data server, but this is exactly where we don't want to stop with this. So we are working together with the Digital Research Alliance of Canada, in the past known as Compute Canada. And they have an object storage system into which all our data is being mirrored into. So that there is like a solid backup of all the data we are collecting, and that includes field data as well as lab data, even though it's not on this slide. Next slide, please. So, and it's not just sitting on the object storage. We have created a data portal. There is a link here to an archive paper which explains the portal a little bit. But I think really the best way to approach the portal is probably through our homepage. It has a couple more images. It has like a document which explains to you how to use the portal and what all the metadata information in there means. So currently the portal hosts two large data sets. The first one is from Eagle Eye. It's about 1.6 million indoor plant images, as you can see down there. So we have like these larger images taken by Eagle Eye, and then we are cropping out each individual plant from it. And the metadata includes also information like how old is this plant, what crop and weed type is it, which imaging angle are you interested in, and each plant also has its own individual ID. So if you like see one plant which interests you, maybe you want to see how it evolves over its life cycle, then you can get only images from that one plant if you want. So. The other large data set on there is the field data, which Chris showed in a previous slide. So you can see here again the tractor with the GoPro mounted cameras on it and the kind of images which are being produced by that. So we call these noisily labeled because not all of the plants in these images are being labeled. Actually, they are not labeled at all, but we know what kind of field we are driving through here and the date at which we have taken these images. So as you can see in the smaller images, there's soybeans, but there's also every once in a while there is a wheat. So this is a managed soybean field. So you know that most of the stuff in there is a soybean. Next slide, please. Okay, so I'm not gonna go through all the details of how the portal works. It's a lot of software engineering happening here. Let me just say it's actually much more complicated to host and surface data in the way we want to. And that is because we don't want the people to like go there and say like, oh, I'm interested in this data. So now I have to download all 3 TB of data and then figure out through the metadata information how to get the stuff I really want. So instead, we have created this client over here where you can make different selections. So in this example, maybe you are interested only in dandelions. You want them to be at most 20 days old, and you are interested in images from an oblique kind of angle. Then you can type that information in here or select it inside of this graphical user interface. And then you get how many files there are. So in this example, it's 3,800. And then you can go and download that, or you can get a sample first to see if this is really what you want. And then you get a random sub-selection of these images. And then you can, if you are happy, you can then go and download exactly those files, and you don't have to go and get like all 3 TB of data or all 1.6 million images. That I think makes the whole thing a lot more attractive and usable for the people out there, and this is exactly what we want. So, next slide, please. Yeah, so we have created this data, this portal a couple of years ago, and we got in contact with the International Weed Recognition Consortium, which is also interested in actually creating this kind of data, especially for weeds, obviously. So you can see here how they work in the field. They are like blacking out the background and find these individual plants, and uh, through a lot of manual labor, they get these images then. And now they are sitting in the similar boat as many others out there. So now we have the data. How are we gonna share it? So yeah, this is how we got in contact because we already have experience with hosting such a portal, and they want something very similar for their lead collections. So it's a good place to collaborate here and make sure that we don't duplicate our efforts. So it could be that their data is being ingested into our portal, or that we are creating a portal, a new one from using ours as a blueprint, so which then will host all the data of the IWRC, but also our data. So there's a lot of options here. We are currently exploring this, but having something like this on the Digital Research Alliance of Canada is very promising because then you know that it will stick around and you don't publish and perish, as Chris said before. Uh, yeah, we are also working together now with Cyber, and I think we have someone from Cyber on the call here as well. So we want to evolve the yeah, there she is, Kate. Hi. So we want to evolve the portal further into the future. So first of all, we have more data and data types currently available, which are not hosted on the portal yet, but we want to get this up there as soon as possible. So we have imaged completely new reads. We have also upgraded the eagle eye images, moving away from the blue background to a black background, having better bounding boxes. We have new kind of field data, like this Fusarium headlight data Chris showed before. The 3D scans and reconstructions is something people are probably interested in, so we want to get all of this on the portal as well. And together with Cyber, we also want to work on integrating cloud-based model training. So the idea here is, why do you want to even download the data when all you're gonna do with it is train your models? So why not just go and train the models right where the data lies at the Digital Research Alliance of Canada? So then you don't even have to download it. And so we want to have like a workflow available on the portal, which allows you to train your models right there and then get the end product, which is a trained model. Yeah, I think that's my last slide. Yeah, okay. No, thank you. So I'm gonna now hand it over to Chris Henry from University of Manitoba, who's gonna talk about these models we were talking about here.
Great, thank you. And thanks for everyone for being here this morning. It's an exciting opportunity to present our work. And so you've seen so far the two of the main pillars of Terabyte. Terabyte fills this niche where we connect with researchers in agriculture, plant science, soil science, biology. We talk about interesting problems that they have that you could solve with creating labeled data sets and machine learning models. And then we go about helping develop automated solutions to solve those problems. And so we have the automated image generation and labeling that Chris mentioned. We host, if we at all possible, we host the data to make it freely available to inspire innovation in the digital agriculture space. And then we're also driving machine learning innovation with this data. So at our Terabyte lab, we do machine learning applications. We also work on machine learning theory. And we make some of those models, as many of them as possible, open, unless we're working with a specific industrial collaboration. And as Michael said, we want to integrate that functionality into our into our portal via a special call from the Digital Research Alliance of Canada. So in terms of our machine learning model development, we create algorithms for specific applications. And in line with our data generation roots, because that's actually how we got into digital agriculture, we're using the state-of-the-art generative methods to look at creating synthetic data to alleviate the most time-consuming and expensive part of machine learning model development, which is creating labeled data sets. So Chris, if you could go to the first slide. This is a problem we've been working on for a number of years, and are currently have a Sustainable Canadian Agricultural Partnership grant to fund. So everyone here probably is aware Fusarium head blight is one of the most significant diseases in cereal crops, especially wheat. And one of the best ways to fight it is just to develop resistant cultivars. And so Chris showed that nursery at Morden, so the nursery at Morden, where we work with Maria Antonia, who does breeding of these resistant cultivars. And he mentioned that she has teams of students that do assessments. She needs to do these assessments in indoor situation and outdoor situation. So what we have developed with the scanner that Chris mentioned is we can develop a method that automatically detects and assesses Fusarium head blight on one wheat head, which was a proof of concept that it's possible. So where we're at right now is we have a few grad students. Some of them are working on developing a push button system working in an indoor environment with the apparatus that Chris showed, with the PhenoSpecs, where we can do multiple wheat heads at once for her indoor trials before she then goes to the outdoor trials. And then we have a few grad students that are then working in 2D and 3D space from data collected on the rover and algorithms eventually intended for the rover to do the severity assessment, the classification and assessment in the field plot trials. And so that's an ongoing project right now. Next slide, Chris. So one of the main applications that we got into at Terabyte was to do weed detection and weed classification. And so we have models now and a set of methodology that if you want to develop an algorithm for detecting a particular type of crop or a particular type of plant in the field, we now have a set recipe that we can share with you on how to go about doing it. And part of it has to do with the synthetic data creation I talked about, and part of it has to do with field data collection. And so we'll come back to that. This is an example of doing yellow foxtail, detecting yellow foxtail in the field, if I recall correctly. And we work with plant scientists on this problem. Next slide. So keeping in the vein of working with plant scientists, we have a collaboration going where herbicide resistance is becoming a big problem. And so some plant scientists have a research program where they're trying to determine which weeds we need to kill so spot spraying is good, but you're still spraying every weed. So you're reducing herbicide application by spot spraying, but it's still encouraging herbicide resistance. So the study is which weeds affect yield and which ones don't. But in order to do that, they're collecting drone data, and they need that drone data automatically annotated or segmented like you see here. So we've been working with them in developing automated methods for the segmentation, and you'll see in a moment ways to synthesize this data so we don't actually have to label it. Next slide, please. I mentioned earlier, we work with all sorts of researchers. We call them domain experts, and we work with them to help expedite their research. So there's some researchers at the University of Manitoba in the biology department that are doing RNAi research. And just from a high level perspective, in this particular example, I can tell you what they're doing is they're adding some of these RNAi sequences to plants, either on the plant or actually within the plant. And when the flea beetles eat that material or try to eat that material, it affects them at a genetic level, and they perish. So where do we come in? Is that what they need to do is they need to assess. Maybe Robert, you could just mute your mic.
The White House that spoke to these outlets. And so you have a president
Or
right
someone
now
could
who
just
doesn't
mute
want
him,
the public
for
or the
us.
media talking about possible decline in health, possible cognitive.
Thank you. Okay, sorry about that. So what they need is they need they have different trials, and they need to assess the efficacy of their trials, and they need to assess the plant material that's left after a certain number of days working with the flea beetles. It's an easy problem if the plants, well, it's an easier problem if the plants are healthy, but as the plants start to wither and die, we still need to separate the plant material from the soil material. And so this is an example of us just finding all the pixels associated with the plant and providing them with a count so that they can do their statistical analysis on which trials are better and which treatments are better. Next slide, please. We have a project with NRC where we are collecting data for the ultimate goal of pea yield prediction and biomass prediction. We picked peas, which ends up being a pretty difficult plant to predict. To predict yield and biomass. So where we've been doing is we have been collecting field data for 3 years with Emily's Innovation Farm. We've also been collecting root data and in order to develop methods for automatically detecting root nodules in the roots. And the goal is that maybe if everyone could mute their mics, would be helpful. And the goal is that we want to see if there is a relationship between we want to develop automated methods to help the plant scientists have a relationship between the roots and the above level biomass. And so, next slide, please. Other things that we're working on are other types of problems. We don't just deep work with, for example, classical image processing models like convolutional neural networks. We work with other types of neural algorithms. In this case, we have we look at graph neural networks and hypergraph neural networks. And so what would be a problem that you'd want to use that for? Well, the Canadian Grain Commission, they have over 20 years of data in terms of FHB occurrence across the prairies, weather station data, satellite data, regional data, soil properties, that sort of thing. And so this information is all interconnected, and the goal would be, can we use it to extract patterns that we can then use to forecast the occurrence of fusarium head blight? And the answer is you can if you start to formulate these things as graphs. However, graphs don't necessarily fit the data model exactly. And so there's these things called hypergraphs, where a hyperedge are these clouds or these triangles that you see, and they actually capture relationships better than just graph data. And so taking this approach, we're working with the Canadian Grain Commission, and we're actually developing ways to forecast fusarium head blight. And our approaches seem to be outperforming the current ones. And so we're going to keep this trial going. And maybe in the future, the FHB forecast will start to be produced based on these hypergraph methods rather than the way that they're doing it right now. Next slide, please. Chris mentioned the photogrammetry system. It's really exciting and have lots of awesome opportunities for not much cost. And so this is an example where we're working with the University of Saskatchewan. They have a hypothesis there that the droopiness level of wheat, so erectophile versus planophile, this droopiness level has some sort of relationship with yield. And so how can they test that hypothesis is they need to grow a lot of this wheat, and then they need to assess the droopiness level, and then from there, arrive at some conclusions. Well, how have they been doing that? By human assessors, and we all know that that's a type of job that humans just aren't very good at, especially over long periods of time. And that's one of the applications that we're working on with our photogrammetry system. Next slide, please. Back to the RNA work. So similar to flea beetles, this is a fungal approach. And just like before, we need to assess the treatments. And we were able to develop a very nice computer vision approach to consistently assessing the size of the leaf blights for preventing this fungus. And this graph here is just showing our relationship in the red of the algorithm with respect to human assessment. And what it is showing is that we have about a 10% error in our model versus humans. But then we ask the humans to go back and do it again, and their re-rating of the same blights was within 10% error. So long story short, they're happy with this method, and it's a useful tool, and it's speeding up their work and their RNA work with this fungus. Next slide, please. A little off topic, but not too much. We also do remote sensing based work, and the remote sensing based work is important because automated agricultural applications will occur at all levels, from satellite to drone to proximal. And so this is a project with the Canadian Centre for Mapping and Earth Observation and Natural Resources Canada. And they have a ton of low resolution digital elevation data from Canada. And they have a little bit of expensive high resolution data. So what this project is doing is developing machine learning methods that will downsample their high resolution data to create a data set of low to high resolution data. And then we're going to develop a second model that can perform super resolution. So taking this massive set of low resolution digital elevation data that they have and actually enhancing the resolution as much as possible to be useful for all sorts of industries available for free from Natural Resources Canada. Next slide, please. Okay, so those are applications, and I just wanted to end the talk with how we are synthesizing data. We noticed early on that data collection is tough and expensive, and if you want to develop scientifically accurate machine learning models, you need scientifically accurate data, and you often can't scrape that off the internet. And when you want to label it, you need expert labeling. It's often that just you can't crowdsource it with the general public. So at the time we started this work, GANs were the prominent or the most popular generative method. And so that's where we started. We took our indoor data that Chris mentioned with the eagle eye. We did some image processing where we extract the images. We would, the plants, color correct it, paste them on top of soil imagery, and then we would use the GANs to make that pasted image look like it was actually grown in the field. And so that was our first attempt, and it was good. And so we didn't stop there. If you want to go to the next slide. So if you remember that first weed detection image I showed you with yellow foxtail in the field, well, the way we developed that model is we took our real field data that we collected at Emily's Innovation Farms, where they allowed us to just let the weeds go wild. We went and cropped a few hundred images of different types of weeds that we're interested. This one is red root pigweed. And then we started using these GAN models to synthesize them. So for one row of these images are real plants, and one row of these images are synthetic plants. And so the spoiler alert is that the top row are the real ones. And we can do the same thing at another type of weed. So that's on the next slide. In this case, it's yellow foxtail. And so again, one row is real, one row is fake. And just to keep the presentation going, the top row is real again. So what can we do with this? If you go to the next slide, what we can do is we can go from a few hundred manually annotated ones that take some time, but not that much time. We can then use GANs to make several thousand fake weeds. And then we can just take, which is easy to do, we can take field data that has no weeds in it, because farmers are very, diligent about making sure they have mostly no weeds, or has mostly no weeds in it. And we can then just insert them using image processing techniques into the image. And due to current machine learning advances, we can know before we put the weed in, we can know where all the plants are beforehand. When we put the weed in, we know exactly where we put it. So we have our labeled data set. And then we can, taking this approach, we can then get several 100,000 examples of weed imagery per weed. So we can easily get, if we're doing 3 weed types, we can get easily, up to like 900,000 images if we wanted to. And that's in the ballpark of what these machine learning models need. Next slide, please. But we didn't stop there. GANs were impressive, but if you go to ChatGPT or Gemini and you ask it to create an image, those sites use something called a diffusion model. And these diffusion models are really good at creating photorealistic images, but you also need a text associated with it, and that's called conditioning the diffusion model. So you can say, I want a panda jumping on a beach ball, and you'll get a picture of that. But what we wanted to do was look at how can we make scientifically accurate and realistic images. And so we take these diffusion models, we train them, we take existing ones because they're trained with sometimes billions of images, so we can get access to these pre-trained ones, and then we can train them with our own strings. And we want to use a string that hasn't been used before. So in this one, we have this weird string, SKS, asterisk, canola with 2 A's, plant, because that doesn't occur in the English language. And by doing that and a few other tricks, we can take our indoor imagery and we can maintain the structure of the plant, but totally generate photorealistic looking backgrounds. And not only just one plant, but we can take that plant and we can add variation and put them into rows, like crop data, like you'd see in the field. So next slide. So I mentioned conditioning these models. You condition them with text, but you don't have to stick with text. You just have to condition it with information. So we're developing ways for that drone data project that I mentioned, where we condition the diffusion model. Instead of text, we condition the diffusion model with image squiggles like you see here. So, and image squiggles are pretty easy to make. So let's say the top one there is you just want rows of soybean with interspersed wheat, which we call the grassy wheat in this case. We condition that into the model, and then because they're great at producing images, if you design them properly, you can get a photorealistic looking drone image out, but you can also get its corresponding segmentation out of it. So now we can synthesize any type of drone imagery that we want, and we can get a perfectly labeled data set out, a well enough labeled data set out that if we have hundreds of thousands of these examples, the downstream model we're training will see through the noise and then can perform the segmentation we want on real drone data. Next slide. And then just coming full circle back to the FHB problem, this is just really initial results, but we feel like the outdoor data problem for the FHB with the rover is also going to be a very challenging data labeling problem. We're working with collaborators from the University of Regina, and they're doing manual annotation. And we think we can also do this synthetic data set creation with outdoor imagery. And so this is just a first pass. It didn't take very long, but we took an image of wheat and we said, well, can we take a perfect image of healthy wheat and just take create some synthetic versions of wheat that have infection rates that we can control, and then superimpose that in the outdoor wheat and then have our labeled data set. And so that's one of the avenues we're trying to pursue right now, to get at a labeled data set so that we can get at a machine learning model that will work to the level of accuracy we need in the field. And then with that, I would just like to show that we couldn't do this work without all the funders that are listed here, and that all these amazing examples, in order to achieve them, we need students at all levels, from bachelor to master's to graduate to postdoc. Thank you, everyone.
Thank you, Chris, Michael, and Chris as well. So I wanted to open it up for questions for this group here. I don't see any in the chat, so feel free to come off mute and ask any of them any questions about the presentation that you have.
We're also happy to meet with people afterwards if you want a more in-depth discussion.
Perfect. And I believe somebody has their hand up, so feel free to come off mute and ask your question.
Gaffa here from Ontario, with management specialist. First, incredible work. Thank you so much. It's really outstanding. But my question is about the synthetic data generation. I'm definitely not from this field. I'm from agriculture, but I imagine that if you're generating that data synthetically, it is based on whatever model has been trained off, but it won't necessarily capture all the variability you will see in nature. So it'll be biased towards whatever image you capture. But is it possible that you're generating images that not necessarily represent the whole range of morphology you will see in the real life?
It's a good question. So you can come at the answer to that question from 2 points of view. So first of all, the underlying theory for these diffusion models is they're really trying to capture the distribution of the data for the type of imagery that you want to capture. So that's just something to know going in. And then, yeah, second of all, what part of this research is entailing is, for a specific application, what's the minimum amount of real data that we need in order to have that distribution captured sufficiently. So I totally agree. Like, if we develop this solution for FHB assessment in Manitoba, And then we go somewhere, I don't know as much about soil, but I imagine you go somewhere where the soil colors are different or something like this, or you go down into the United States where maybe they have, different lighting conditions.
Mhm.
Maybe the accuracy drops. But in that case, we have the set methodology where, look, it worked in this geographical region that we want it to work. We collected X amount of images. Once we had those X amount of images, we had a model that reaches, for example, 95% accuracy. We just need to collect a little bit more data in the field. The whole point is what's the minimum we can take to alleviate this manual data labeling aspect. So I agree with you. Any new machine learning problem that you start to tackle, these are the questions you need to answer. How much data do we need? What sort of biases will there be in the data set? So yeah, I totally agree with you.
Okay, and I believe Anne Michelle is next to ask her question. And Michelle, are you there?
Could you hear me now?
Yes, perfect. Thank you.
All right, sorry, between all this technology. Sorry. So Christopher, I was saying your presentation is super interesting. And you mentioned NRCan and AFC. So it's just to confirm that you're working with as well the right people within our department. I am from Agriculture and Agri-Food Canada. And we have now a team about 15 people working on quantum and machine learning and algorithm. And we have this project using our satellite data to mix with meteorology data with soil data and draw, to come up with new models of farming. So you are at a lower level, which is at the plant level. So I hope my colleagues talk to you about this project that we do on quantum, because otherwise connect with me and I will connect them with you.
Sure. Yeah, that'd be great. Like one thing that we've known for a while that we just haven't gotten to is that agricultural problems are multi-scale. And I think there's like a lot of interesting work to be done with projects that have like this tipping and queuing model where you start at the satellite level and extract some patterns or make some decision, go into a field at the drone level, do a wide scale scan of the field, again, extract some patterns, make some decision and then maybe take some action at the proximal plant level. And so we have satellite experience in this team. We've done machine learning models with satellite data, for example, for land use, land classification. We just haven't tied the satellite to the drone to the proximal because we can't do everything. So if your team is doing satellite, I think there's lots of opportunity to look at what's some sort of multi-scale analysis that we can work together on.
Exactly. Yeah, because NRCan and AFC, we have access to the same data sets from the satellites. So I hope my colleagues talk to you about that. So anyway, I am on Geds. I don't know if you can find me, but otherwise the lady from with can connect with us. But do not hesitate to reach to me.
Great, yeah. And where are you located? Because our Enercan folks are in Sherbrooke.
I'm located in Ottawa. The
Okay,
team working on quantum is in Sherbrooke, next to Sherbrooke, because this is a quantum hub, University of Sherbrooke.
okay,
But they work with Gagari University, which is another quantum hub.
okay, okay. Well, next time we're at Sherbrooke, we should come and meet in person.
Exactly. So let's connect. Thank you.
Great, thank you. Perfect.
Yeah.
And our next question is from the chat. Vinay was asking that all this data, is it only for research use case, or is there commercialization mode to use these images for integration with other softwares?
Michael, you remember the exact license? I'm pretty sure. Well, if you remember it, my recollection is we released it under Canadian Commons, or yeah, no, not CC. It's like CC BY. I can't remember the specific example, but short answer is yeah. It's there to spur innovation in the agricultural sector. If you publish a paper, we would like some citations, but otherwise, we want people to use the data.
Perfect. That sounds
Yeah,
great.
it's a it's a pretty straightforward process, I would say. You go to the homepage, and then you get the client, and you write me an email, I make you an account, and then you will be able to download the data.
And I think I said Canadian Commons, I meant Creative Commons. So
Perfect. David, you have your hand up next.
Yeah, I'm going to say something a bit contentious. I would suggest that you limit this database a little bit. I know you're academics and you love to share, and let's share data. And I know open source works for innovation, but at the same time, I would suggest that you establish formal collaborations with everybody that wants to access your database. And that criteria might be they're Canadian, or they're Canadian companies, or they're Canadian researchers. You can always open up your database later, and or if they're international, as long as they collaborate with you. Let's make sure, I know this is not what academics want to hear, but let's make sure that it's a bit of a quid pro quo, that you're giving them data, they're giving you something that's useful to you too, so that we both can benefit. And I think sometimes Canada is a little too nice. We do a little bit of innovation philanthropy where we do stuff and we give it to the world, but the world doesn't necessarily give us a lot back. So I think collaborating across Canada is a fantastic idea because we're a small country and every university in the ag space should be working with you and us. We're University of Guelph, so that we have global scale. But let's get our shit together first before we give it away. I think that's really important because data is the new intellectual property that is going to identify us in our role in the future. So just because they email you, be a little bit more resistant and scrutinize who they are, because there's going to be lots of players, including those in Asia, that they're going to scrape whatever they can get for free, and it's not I just really worry about our competitiveness if we're doing this and we're not establishing formal relationships. So you might already be doing that, but that's my point.
I can address this. So this is why in the process I just described, you're gonna have to write me an email, and then I make you an account, and you will get the access, right? So if Google knocks on our door, I will not make Google an account, and it's not scrapable, right? So without an account, you will not be able to get any data. So we are looking at who is knocking at our door, and but I think we are pretty much aligned here, as you said, Canada-wide, researchers, definitely yes, Canadian industry, yes, but yeah, we want to share it, we want to spur innovation, but we are still doing like a sanity check on, okay, like who is actually coming here.
yeah, and I agreed. Thank you, David. That's a great comment. And the funding that originally got the portal off the ground and collecting all this data was from Western Economic Diversification, which is now Prairies Canada. I can't remember their new name. Sorry, it's escaping me. Part of that project had a data management plan where the first few years only researchers and then industry, and then we make a decision. And we've been at this for a long while, so we've gone through both those embargo periods, and now we just do as Michael says, we do it on a case by case basis.
And maybe I'll just add, you'll be happy to know that we have shut the door a few times. So there's a plant app out there called Picture This, and you take a picture of your plant and it tells you what your plant is. And lots of people ask us, why do you need to do your work? Because this has already been done, because I've seen this app. But of course, that app is limited to certain cases and certain ages of plants. And they're never going to share their data with anybody. But interestingly enough, they came to us asking for our data, but we did not give them our data. So we are being discretionary about this. But it's a very good point, David, and something we should all heed. So thank you.
Perfect. I think we have one more question in the chat. Do you require large data sets to train your models?
That's the million-dollar question whenever starting a new machine learning development problem. It can range from a few in the hundreds to 100,000s to over 1 million. And it depends what you're trying to do. So these pre-trained generative models, especially the diffusion ones that create the realistic imagery, in this case, you can modify the architecture and still retain a lot of the information. And you only need on order of like hundreds to create synthetic data. So the drone example I showed you was just using on order of hundreds out of about 5,000 images that we have, real field images, excuse me, that we have. The one that you saw where with the GAN approach, where we cropped some images from the field. So again, it was a pre-trained GAN released by NVIDIA, and that only needed on order of hundreds of cropped images. And we do one model per weed. So we take like 300 cropped images of red root pigweed, and then take this pre-trained model, train it, and now we could easily generate, 3, 4, 5,000 examples with variation. And then if you have 3 or 4,000 examples, and then you take outdoor field imagery that's weed free, you can easily generate 100,000 examples by taking just weed free imagery, randomly sampling from your few thousand weeds, doing some random like rotation, flipping, that sort of thing, randomize the amount of weeds in the photo, and it's easy to get up to the scale of 100,000. And so where you need large amounts of data is then for this downstream task. And again, it's the million-dollar question. How much do you need? But typically when you're doing these real world problems, out in the field under varying conditions that was mentioned before, you're going to need, data sets in the tens of thousands to the hundreds of thousands, but it really depends on what you're trying to do. That leaf blight, Chris, if you want to bring up those leaf blight examples with Xavier's work, this is a very constrained problem. So in this case, they're taking pictures on a white background. And so here, the machine learning models, they're not having to sift through all these real world conditions. They're basically getting a green leaf on a white background with a blight in the middle. So here we needed several hundred labeled human examples. So it really comes down to the problem, how difficult it is, how much variation. Like if you look at the ImageNet challenge that spurred this all on in the machine learning revolution 2012, it was 1,000 categories like bus, cat, dog. 1,000 categories in the training set was 1.3 million images. So it ranges.
Maybe to add to this, I think the society is getting a little bit nervous about when it comes to data sets. So if you look at articles out there and what people are saying about large data sets, so I think large data sets can mean a lot. You can also when you think about large data sets, you can think about like the kind of data sets with ChatGPT or something similar is being trained on, where people go and like build entire data centers. So we are very far off of that here. Even our 1.5 million images, which we have on our portal, is not even a drop in the bucket if you compare to this kind of what you might mean as large data sets, right? So there's a wide variety of scales here, but when we say large data sets, so as Chris said, some of these models are being trained on hundreds of images, others like maybe on tens of thousands or hundreds of thousands of images. Um, but all of these images are still fitting on still a normal computer or on our on our servers, right? So we certainly do not have a data center sitting somewhere which hosts a ChatGPT level of data or something similar like that.
Great. Okay, I think I'm going to cut off the questions because we only have a few more minutes left. So thank you everybody for your questions and the discussion. Again, thank you for our speakers for joining us today and sharing their research. They did share their contact information in the chat as well. So I definitely encourage everybody to continue this conversation and reach out to them with your specifics as well. And then Anne Michelle, she also shared her contact for Chris as well to reach out. Um, and then I just have one more introduction that we have. So Kate, you invited Ron Van Holst from Compute Ontario to join us today. So Ron, if you just want to introduce yourself, and then we can close out today's meeting.
Thank you. Yeah, my name is Ron Van Holst. I'm with Compute Ontario. I really enjoyed this talk. I have somewhat of an agricultural background, so I appreciate it very much. I'm a Guelph native, and I have many family in farming. But I'm more of a data nerd, more of a supercomputing nerd. So the research here is amazingly fascinating. Two questions. First of all, right now I'm focusing on research security for Compute Ontario. That's a national priority, and I'd like to ask from the researchers their research, their data collection, and the comments on data sharing and Canada being too nice. I heard that, wrote that down. How the research security requirements from ISED will impact your research data curation and sharing.
Okay. Actually, if those are new requirements from ISED, I'm not aware of them. I could just talk about, most of our data collection has been curated with the idea of sharing it. So from that point of view, the data security is not really important, but we do have collaborations like with companies like, for instance, MacDon. And in those cases, the data that they give us, it has to sit on its own server in our lab, and only like one person has access to it. Once the project's over, we have to destroy the data, and we have a data management plan and a protocol for that. A lot of colleagues work with medical data in my department, so they have to make sure it's housed, on Canadian servers that have special access. Because we go the open data route, we don't have to necessarily have, those secure protocols in place. But maybe you can update me on what are the new ISED requirement? Are they new?
Well, I don't think they're not that new. You
Okay.
did comment on the you did contact on the comment on the data sovereignty, which I think is very good.
Yeah.
So I'm glad you did. Not enough people are talking about that, but I think in Canada we need to take our data sovereignty much more seriously. So I'm glad you opened that subject. No, I'm just wondering under ISAID sensitive technology research areas. I don't see ag tech specifically, but your work with advanced machine learning to process the data. You work with sensing, advanced sensing,
I see.
and even like genetic engineering
Yes, yes,
to
yes.
get around, crop sensitivities and resilience to certain diseases. I'm not an expert, but all of those things touch on sensitive
Yeah,
technology
yeah, I
research
understand
areas.
sorry, I misunderstood
And
the
it's not
question.
just data leakage, it's also, foreign entities maybe trying to contaminate your data and falsify your results. That's a
So,
risk factor.
sorry, I can respond now. I get I misunderstood the context of the question. So yeah, any funding that we get, we have to mention if we're doing research in a sensitive area. Machine learning is one of them. Large data sets is another. So we have to have a plan in place to and dual use of technology. So for all of those things, we have to have a plan in place. We have to comment on it. And so yeah, we have to make sure like we have a method for addressing if our technology, or our data is being used by foreign entities, and there's a list that Canada publishes uh,
Yeah.
that you have to be aware of. So those things are in place, and their institutions help us make sure that we meet those requirements. In terms of our data, it's all contained in Canada. There's one server at U of W that Michael mentioned, and then the rest of the data is on the Digital Research Alliance of Canada. So it's definitely all within our border.
Okay. Yeah. Thank you very much. You mentioned the dual use as well, which is a hot button right now. The sensing technology you're using could be, factors of dual use. Thank you. Thank you very much.
Perfect. So we are over time. So that was great discussion. And then Dan will be back next week to continue hosting. So thank you again to our speakers and everybody who joined today, and have a great day.
Thank you.
Thanks.